The overall goal of this procedure is to incorporate an ABC transporter, in this case the maltose transporter MAL-FGK2, into soluble membrane nanodisc particles. This is accomplished by first mixing the membrane scaffold protein, MSP, with the transporter and the correct amount of lipids in detergent micelles. The second step is to add polystyrene beads and to gently rock the mixture overnight. The detergent is progressively adsorbed onto the beads, allowing the formation of the nanodisc particles. Next, the beads are removed by sedimentation. The final step is to separate the properly formed discs from aggregates and unassembled scaffold proteins by gel filtration chromatography. Finally, the quality of the discs is assessed by native gel electrophoresis or light scattering spectroscopy.
The main advantage of this technique over other methods like liposomes is that membrane proteins in discs are perfectly soluble in aqueous solution.
In addition, the protein domains are now accessible from both sides since there is no more membrane compartment.
Using membrane scaffold proteins of different lengths, I was able to trap and study the different oligomeric states of membrane proteins.
Most importantly, this method can help answer key questions in the membrane proteomic field, such as the identification of novel binding partners of ABC transporters.
In fact, the implications of this technique extend further toward therapy, because it can help the discovery of activators or inhibitors of membrane proteins and receptors.
Though this method seems simple, caution should be taken when choosing the correct protein-lipids ratio.
Generally, individuals new to this method will struggle in calculating the correct molar ratio or doing the correct control that ensure the quality of the preparation.